So the next speaker is Erin Getty from Queen's University, Belfast, and she's going to be talking about BA.1 induced antiviral response attenuating RSV infection.
Thanks, Isabel. As she said, my name's Erin, and I'm from the Wellcome Wilson Institute for Experimental Medicine at Queen's University, Belfast. I'm a second-year PhD student, and today I'm going to be presenting our work on RSV and SARS-CoV-2 co-infections, which were alluded to a little bit yesterday. So I don't need to introduce SARS-CoV-2 to this audience, but what the pandemic did do was highlight respiratory viral infections that are of major importance to public health, and this includes RSV. RSV is a single-stranded negative-sense RNA virus that was first isolated in the 1950s from chimpanzees, and then later as the infectious agent of severe lower respiratory tract infections and in infants. Indeed, RSV causes hospitalization in children under the age of 5, and this equates to about 3% of hospitalizations in infants. It's also a major cause of lower respiratory tract infections in the elderly, as well as immunocompromised individuals. So there's a significant burden to both ends of the population spectrum. So like SARS-CoV-2, RSV is circulated in large respiratory droplets and through close contact. And RSV in particular typically circulates in the winter months between November and February. However, due to the mitigations and lockdowns to control the spread of SARS-CoV-2, there was a complete elimination of RSV circulation in 2020 and 2021, as you can see from the graph here. And then an out-of-season surge in 2022, as you can also see from this graph. And this offered the opportunity for co-circulation in both viruses with unknown outcomes. So as both RSV and SARS-CoV-2 target ciliated cells of the respiratory epithelium, we aim to investigate virus-virus interactions and the host immune response to these co-infections using our well-differentiated primary airway epithelial cell model, which I'll refer to as WD PBECs for the rest of this talk. So in brief, what is our WD PBEC model? So we take either primary nasal or bronchial cells and culture them in monolayer, like a cell line. Once these reach confluency, we transfer them to transwells. Again, these are submerged until they reach confluency, and then once this happens, we remove the apical media to create an air-liquid interface, which then causes differentiation to our pseudostratified epithelium, which then allows us to mimic the airway epithelium in vitro with great authenticity to the airway epithelium in vivo. And hopefully, if this video works, you can see the ciliated cells beating in our transwell model. So if you draw your attention to the top corner, you can really see the cilia beating. So it's a really nice model to use. So using this, we have 3 infection schemes. So we have a single infection with RSV, the clinical isolate BT2A, or a single infection with BA.1 SARS-CoV-2. We also have a simultaneous infection where we infected our WD PBECs with both viruses, or then a sequential infection scheme where we infected with one virus and then 48 hours later infected with the second virus. So does RSV have an impact on RSV growth kinetics was our first question. To determine this, we measured infectious titers by plaque assay on VITs. To date, most of the co-infection studies have focused on the simultaneous infection model where both viruses are added at the same time. So initially we looked at this. As you can see, there's a trend for a decrease in SARS-CoV-2 infectious titers. However, this isn't significant, and it's likely due to the donor variability that we see when we use our model. Then moving on to a sequential infection scheme. So in this case, we infect first with SARS-CoV-2 and then followed with RSV infection. And at 96 hours post-infection, you can see there's a significant reduction in SARS-CoV-2 titers. Then looking at the other way around, so what happens when we first infect with RSV? Again, there's a trend for a reduction in SARS-CoV-2 titers However, this isn't significant, and it's likely, again, to the donor-donor variability that we see using our model. Now, this is where I'm going to probably leave SARS-CoV-2 and talk about RSV, because that's actually the virus that I'm most interested in. So again, we looked at, does SARS-CoV-2 infection have an impact on RSV growth and kinetics? In our simultaneous infection, there was no difference in RSV titers. Similarly, that's so difficult to say, when we infected with SARS-CoV-2 following RSV infection, there was no difference in RSV titers. However, what was interesting to us, and our group, was that when we infected WD PBECs with SARS-CoV-2 prior to RSV infection, there was a significant reduction in RSV titers at 48 hours post-infection. And this time point, I'll point out, an early time point, seems to be the critical point in determining the outcome of infection. So we want to identify a mechanism for this, and as we all know, the antiviral response is really important in determining the outcome of viral infection. So initially we used RT-qPCR at 48 hours post-infection to look at the expression of the type 3 interferon, interferon lambda-1, as downstream ISG, so ISG15, MIX1, and DDX58, otherwise known as RIG-I. As you can see, irrespective of infection, there's an induction of an antiviral response, and this was also replicated at the protein level with secretion of protein as measured by ELISA. However, while the type 3 interferon response is considered most important, especially to respiratory viral infections, of interest to us, and also shown by a recent study, interferon beta upregulation at 48 hours post-infection has been shown to block influenza infection at this early time point. So therefore, the upregulation of interferon beta by SARS-CoV-2 at 48 hours post-infection could be the potential mechanism for the reduction in RSV titers that we've seen. So to further elucidate if this is the case, we set up a conditioned medium experiment where we infected our WD PBECs with SARS-CoV-2, collected the basal lateral medium, transferred this to new WD PBECs from the same donor in the presence or absence of ruxolitinib, a JAK-STAT inhibitor, and then infected with RSV. So as you can see from these growth kinetics, when we blocked the JAK-STAT signaling pathway, we saw a restoration of RSV growth kinetics, and this was significant at 48 hours post-infection, which is the time point that we think is critical to determining the outcome of the co-infection. As you all probably know, ruxolitinib is not specific. So work to date using our cell lines, Bs2Bs and A549s, to try and determine which interferon is driving this response, where we treated the cells with recombinant interferon beta and interferon lambda-1 suggests that interferon beta is better at attenuating RSV growth kinetics. I'll note the concentration of interferon beta that we used here is comparable to what we are getting released from in our conditioned medium from our WD PBECs. So I hope today that I've shown that prior SARS-CoV-2 infection of WD PBECs attenuates RSV, and this is likely mediated by interferon, primarily interferon beta, with the data that we have to date. And hopefully this means that there is a less likelihood of exacerbated RSV-associated disease. Future work involves confirming the functionality of interferon beta by use of neutralizing antibodies, or if anybody has some of the HEC reporter cells for interferon beta and would like to provide a file, that would be great, so we can know how much interferon beta is being produced apically and basally from our WD PBECs. That would be great. Just to bring it back into this session, I'd also like to point out that this work is really important in understanding what happens in co-infection setups in terms of when is appropriate time to deliver vaccines, as if the epidemiology of RSV seasonality is going to be impacted by SARS-CoV-2 continuous circulation is important to understand. And while a few months ago I would have would have stood up here and said that RSV remains an unmet medical need due to the lack of prophylactics and vaccines, I can now happily say that in part due to the SARS-CoV-2 pandemic, 2 vaccines have recently been approved, both a maternal vaccine and a vaccine for the over 65s, and a new monoclonal antibody to be given prophylactically has also been approved. With that, I'd like to thank all the members of the Power Lab, both past and present, especially Dr. Lindsay Broadbent. I'd also like to thank all the funders and the UK ICN for providing funding for me to come and present to you today. Thank you.
Thank you very much, Erin. That was a great talk. Does anyone have any questions? Yep.
Not to Holger. If there's an early career person that wants to ask a question, you can go first. No? Okay, I'll take it. So very interesting, and the mechanism that you've identified is great. Just to say, we had made some observations when we were performing some LAVE vaccine studies, then challenge with SARS-CoV-2. I think we published it, but if we didn't, you do know it. Okay, so but we never worked out the actual mechanism. I was wondering if you put any live attenuated vaccine, the BA.1, on your cells to see if we're getting into the beta and alpha being produced.
We haven't yet, but it'd definitely be something to consider moving forward with our work.
Are there any more questions? Okay, great. Thank you very much.